Hi, welcome back, everyone. I hope you enjoyed some posters and chatted with each other. Myself looked around. Looks really good, actually. I'm excited to continue with the last session of today, day one. And this session is going to be about a very important topic that's very near and dear to my personal heart, which is scientific modeling of VHL. And, I think that we know, well, those of you who know that my research career was based on modeling VHL disease. We developed a zebrafish model to that end, as well as cell models. And it was something that I think really added value to the research, but also the translational aspects of the disease. And it's a tough disease to model, let's face it. It requires lots of different tissues, and the mice have not been very easy to generate. So I think that's really brings us on to the first topic, which is going to be Ian Frew, Professor Ian Frew from Freiburg in Germany. It's near the south. It's near the Swiss border. It's a beautiful place. If you ever go there, look Ian up. And Ian, I'm going to give you this opportunity to tell us your story.
Thank you very much, Rachel, and thanks also very much to the organizers. I'm just trying unsuccessfully to click on the screen sharing icon. I don't know why that's not working. Yeah, I'd like to, I'd ideally like to do it for my own slides, because I changed a few things actually since I sent you the last version. For some reason, camera, the buttons are slightly offset. I don't know how to do that. Okay, we might have to go with your version, Eileen. Sorry. Everything else has worked perfectly. I'm still trying to get mine to work. Okay, so does everyone see the slides? Maybe you can put them in full screen mode.
Presentation mode, please, I think.
Presentation mode.
Thank you.
Okay, so thanks very much again for the invitation to speak here. Sorry for the technical complications. So what I'd like to discuss with you are actually two things. One is to very quickly fly through a recent publication that we had, and I'll really go over this in brief, because there's more information, of course, in the publication, but just to tell you the key messages about the role of HIF1 and HIF2 and what we've learned about those in CCICC through mouse modeling. And in the second part, I'd like to tell you about how we're trying to use those mice to gain some therapeutic insights. So I need the next slide. And if you just click through all of to the end, yeah. So, sorry, this is not going very well, but the basis of the idea that we're talking about was introduced very nicely by Bill Kalin, who gave a very nice overview of the role of the different potential roles of HIF1 and HIF2 in CCICC, with HIF2 acting to promote growth and HIF1 rather acting to restrain growth. And there's a lot of evidence that's been presented, very good evidence, of course. But what we have to note about this is that all of these analyses are done on kind of end-stage CCICC, so in cell lines, in mouse xenografts, or in analysis of human tumors. And we've not been, basically by definition, able to address the role of HIF1 and HIF2 in the actual the full process of tumor formation in situ. So if you have the next slide, we decided to look at this using our recently generated mouse model that was based on the deletion of VHL P53 and RB, a somewhat artificial setting, but nonetheless it seems to quite accurately model many of the aspects of CCICC. So these mice develop tumors after a certain latency. We're able to monitor those with micro-CT imaging, which you see at the bottom panel, which is very helpful for the therapeutic studies I'll tell you about later. And you see in this Kaplan-Meier curve on the right, showing you the effect of VHL deletion in the P53RB background, that it indeed accelerates tumor formation. And if we jump to the next slide, what it in fact does is it induces the classic features of clear cell renal cell carcinoma, namely that we have a range of different cytoplasmic morphologies, ranging from really the clear cell phenotype through to a more eosinophilic phenotype. But importantly, what we see is a very dense and intricate vascular network that you can appreciate from the CD31 and the von Willebrand factor stainings. And in the bottom panels, another nice feature of the mouse model is that it reproduces many of the precursor lesions of CCICC that we see in VHL patients, namely the cystic lesions that then can grow, these atypical cysts that then can form tumors, as well as small solid tumors that appear to precursor tumors of bigger solid tumors. So we decided to take advantage of this mouse model in a very long-term project. If you jump to the next slide, please. And we introduced now HIF1 and HIF2 deletion into this background to try and address this question of the different role of HIF1 and HIF2. And I've used shorthand here where VPR indicates the VHL P53RB gene deletion. H1 is then the HIF deletion on top of that, and H2 is HIF2 deletion. These are inducible epithelial cell-specific deletions driven by the KSP-Cre promoter. And what you can see very nicely from this huge experiment that was over many years and summarized really quickly in this one slide is that the deletion of HIF1, the blue curve, completely takes away the tumor-promoting activities of VHL, whereas the deletion of HIF2 rather has an intermediate phenotype. And we see the same thing in panel B when looking at the number of tumors per mouse, that there's a complete rescue with HIF1 deletion and a partial rescue with HIF2 deletion. And to a lesser extent, the growth rate of those tumors. So this was really somewhat surprising in a way, at least in this genetic background. HIF1 seems to be the gene that is really most necessary for the evolution of tumors, right from the very first day, if you like, of tumor formation. So next slide. Can we jump to the next slide, please? Yep, wonderful. Oops, go back one. Okay. So I see this a little bit blurry, but no, now we've gone forward. One more, go back to the immunohistochemistry slide. Yep, thank you. So this slide was a confirmation slide or a confirmation analysis just to basically prove to ourselves that HIF1 and HIF2 were deleted where they should be. This is important because we're talking about a rescue. We need to know that the tumors have actually arisen in the absence of the genes and not that there's some escaper effect. And indeed, we saw that HIF1 immunohistochemistry was disappeared in the VPRH1 tumors, and HIF2 immunohistochemistry had disappeared. The nuclear staining had disappeared in the VPRH2 tumors, but not in the other genotypes. So indeed, the Cre activity was doing what it should have and deleting all 4 genes at the very onset of tumor formation. Now, we looked very carefully at the histology of these tumors and for phenotypic differences, and what we didn't see very many differences. What we did see, however, was a shift in the balance of the so-called clear cellness of these tumors. So I mentioned before that the VPR tumors showed different histological appearances in terms of the clear cell appearance. So clear cell score 1 on the left is really completely clear cell through to intermediate clear cell, and then a more eosinophilic appearance. And what you see in the bar graph is a quantification of this, where about 60% of the tumors in VPR have a degree of clear cellness, and this is then shifted when we delete either HIF1 or HIF2 to the more eosinophilic appearance. So if we jump to the next slide, I can discuss what we think are the reasons behind this. Yep, wonderful. No, skipped over one. Yep, thank you. Can we go back one to the blue and yellow slide? Thanks. So this is a lot of information on this slide. This is a summary of the insights we gained from RNA sequencing, and there are basically 3 points I'd like to make here. In panel B, you can see that as we expected and predicted, that the glycolytic gene expression signature is dependent on HIF1, but not on HIF2. And we think this may play a role to explain the role of HIF1 in the clear cell phenotype. And in panel F, one sees that there is a HIF2 dependent signature that's rather involved in lipid and cholesterol metabolism that may also play a role in the clear cell phenotype. And it may be that these 2 different functions contribute or are both necessary in achieving the different metabolic control. The surprise we got out of these analyses was in the HIF2 deletion model that a lot of terms associated with T cell activation or interferon signaling or antigen presentation were highly up or were more upregulated on a gene set enrichment analysis in the HIF2 knockout than in the other 2 knockout genotypes. And now if we go one slide forward, please. this is one of the reasons we think for this was the analysis that we did of the tumor immune microenvironment. And this is now a whole series of quantifications focusing on the boxes here in red in the CD8 positive T cell compartment. These are enumerations of stainings actually of the tumors. So these are not just RNA expression values, but real quantifications of immune cells in the tumors. And what we noticed was that in both the HIF1 knockout and the HIF2 knockout genotype, there was an increased T cell infiltration in both of those sets of tumors. But when looking at markers of T cell activation, CD69 and perforin in panels E and F, we saw that only in the HIF2 knockout was there actually an upregulation of these activated T cell markers. And this didn't occur in the HIF1 knockout. So going forward and building upon this, if we go to the next slide, we wanted to ask whether this has a correlation in human tumors. And we did the best thing we could do, which is to take advantage of the TCGA data sets, where it's known that loss of the chromosome armor 14q that harbors HIF1 alpha occurs in about 40+% of CCRCCs. You see that represented in blue in panel A, whereas HIF2, if anything, tends to be gained. And it's known, of course, in panel B, this is not new, but HIF1 alpha deletion correlates with a poor outcome of patients in sporadic CCRCC, whereas gain has no impact. And when we looked at the correlation now of HIF1 alpha loss with the RNA expression levels of markers of the genes encoding the CD3 receptor of CD4 and CD8, you can see on the left panel of the graphs that the HIF1A loss tumors indeed had, on average, about twofold higher expression levels of these T cell markers, which is consistent with what we saw in the mouse model. And on the other hand, HIF2 gain had absolutely no effect in this assay. Going forward again, one slide, please. This is an analysis, a much more complicated analysis based on bioinformatics. The panel you see on the right, the columns are representing different comparisons. So the first comparison is HIF1 alpha loss tumors versus unaltered. And each of the rows represent different gene expression terms that are characteristic of specific immune cell subtypes. And these analyses are looking for the enrichment of these particular subtypes or these particular RNA signatures in the comparison. And what this analysis showed was that, again, the HIF1 alpha loss tumors show a very broad spectrum of changes in different immune cells that are indicated there, with yellow indicating upregulation or an enrichment of those putative immune cells, and blue representing downregulation. The HIF2 gain versus unaltered showed almost no differences, but what we did notice was that when one looks at the mRNA abundance of HIF2 alpha in CCRCC, the KIRC there, you see that there's a very wide distribution of HIF2 alpha expression levels in CCRCC tumors, whereas HIF1 on the left shows a very narrow distribution. And when we looked and compared now the high expressing HIF2 to the low expressing HIF2 in the third panel of panel N, you also see that there are very robust expression or robust abundance differences in the predicted abundance of different immune subtypes. And I won't go into more detail there, but this we think is indicative of an altered immune microenvironment. So going to the next slide, I'd like to just conclude this section. As I said, this is all published, so I've rushed through it, with a couple of caveats to start with. One, of course, any conclusions we draw here may be contextual to mice and not necessarily reflect the state in humans. And it also, of course, may be contextual to this particular genetic background that we've used. Even though it reproduces many of the features of CCICC, this may represent one subtype or none or all of the subtypes. We don't really know. But taken at face value, I think what we can conclude, at least, is that HIF1 in this setting is definitely necessary for tumor development, whereas HIF2 alpha is rather largely dispensable. And this is kind of the opposite of what we might have predicted starting out. We also showed, very interestingly, that the HIF2 deficiency in the mouse CCICC cells increases antigen presentation, which I didn't show you, and also markers of immune activation. And finally, that at least in humans, the copy number of HIF1 alpha and mRNA abundance of HIF2 alpha correlate with altered immune microenvironments in CCRCC. And this is something we may potentially have to think about when stratifying immune checkpoint therapies. So building on these two findings in particular, namely the HIF2 dependence and this idea of potentially altering the immune microenvironment, we wondered whether we could do or learn something more therapeutically about studying this mouse model. So if we go forward one slide, of course, Bill Kalin introduced this very beautifully already, and his lab, as well as the lab of Jim Brugarolis, have pioneered the studies of this together with Peloton, where they beautifully showed that in preclinical models there are excellent responses in some cell lines, and this is exemplified by this study from one of the Nature papers of the PDX models, where many of the PDX models were sensitive, but equally interestingly, some of them were insensitive or showed intermediate sensitivity. And I think if we go to the next slide, this pattern is now being borne out in the clinical trials, which are extremely encouraging, and I don't want to disparage them in any way, but there are certainly patients that are responding well, some that are responding quite well, and others that are responding less well. And so I think there will be the case, and it seems to be emerging, that there are some tumors that are resistant. And so we decided to test HIF2 inhibition in this model that I'll come to in a moment. But we also tested two other drugs, and I'll introduce those to you in the next two slides. If we go forward one, please. This is a drug that was actually rediscovered. It's a very old drug. It's an old antibacterial agent that was used all the way back in the early 1900s. It was rediscovered by the Semenza group, acroflavin, to be an inhibitor of HIF1 and HIF2 dimerization to ARNT, so thereby blocking both the activities of HIF1 and HIF2. If you jump forward now, another slide, this is one of the drugs that we've tested. Skip forward here. The second drug, actually, if you click through all of the animations to make it easier, that would be great. Perfect. Stop. Oh, yep. Thank you. The other drug that we're interested in was already alluded to, actually, in the wonderful talk earlier from the Zhang lab, who discovered that sphingosine-1 kinase was an important target of the transcription factor SFMBT1. And sphingosine signaling is interesting. It had previously been implicated in CCRCC because this signaling pathway was implicated upstream of HIF1 alpha, and the G protein coupled receptor that this pathway is signaled through activates mitogenic signaling through RAS and PI3 kinase. And this pathway basically works by converting sphingosine lipids into the soluble messenger sphingosine-1 phosphate, which is then excreted from the cells or transported from cells via these specific transporters. And then these act on the sphingosine-1 phosphate receptors. The reason this is interesting is because there is a drug which is FDA approved. This is Fingolamod, which is actually an antagonist or a superactivator, in fact, or it basically downregulates sphingosine signaling. This is FDA approved, and it is given to patients with multiple sclerosis, orally available. And as previously alluded to, sphingosine kinase 1, you see on the right-hand panel, is upregulated in CCRCC compared to normal. And we see the same thing in the mouse model. So these three drugs listed here, acroflavin inhibiting both HIF1 and HIF2, the Peloton compound inhibiting just HIF2, and Fingolamod were the three that we tried in clinical trials in our mouse model. If we jump forward now to the results, here you see again really several years of work summarized again in one simple slide. I think so. Three more slides. You see here on the left panel the tumor growth curves, and the tumors in this model grow exponentially, which is somewhat different to human CCRCC. And these growth curves have now been normalized to the day of treatment initiation. Mice were treated with acroflavin for a short period of time with the HIF2 inhibitor, PT2399, and also with Fingolamod. And what one already instantly sees is that Fingolamod, at least in the short term, causes tumor regressions and stabilization of growth. And this is all quantified now in terms of tumor growth rate over the first couple of weeks of therapy in the bottom graph, where you can see that acroflavin has a therapeutic effect, albeit a relatively modest one. The Peloton compound, disappointingly, at first, did absolutely nothing in this model, but Fingolamod showed some nice therapeutic responses. So we go forward one more slide. This is just an example to show that we think we at least are hitting the targets, at least in some of the tumors, immunohistochemistry for HIF1 targets, CA9 and GLUT1, at least in some tumors, acroflavin and Fingolamod, FTY720, show downregulation. This is actually typo, it should be PT2399, not 2385. But those mice don't actually show inhibition. So at least we're hitting the targets we think we'd like to hit. Skipping one slide forward, please. Phenotypically, we do think we see an on-target effect of the Peloton compound. This is now looking at the histological appearance of the tumors. The first three columns are showing you exactly the same data I showed you before, that HIF1 and HIF2 deletion cause this more eosinophilic appearance. And we phenocopy this largely with acroflavin, fingolamod, and Peloton. So we seem to be at least having probably metabolic effects in these tumors, but if not therapeutic effects. The final slide I'd like to show you is now looking at the immune microenvironment. And as I said, we're very interested in the fact that HIF2 knockout seemed to promote anti-tumor activity, at least by CD8 positive cells. And so we thought maybe by mimicking this pharmacologically, we might have the same effect. Keeping in mind here, of course, that the pharmacological approach now inhibits HIFs in all of the different immune cell subtypes, where they certainly play an important role. And indeed, what we saw, if you focus on the CD8 positive T cells, was that PT2399 slightly decreased CD8 T cell infiltration, largely decreased B cell infiltration, as well as macrophages, and Fingolamod inhibited macrophage infiltration as well. So this was not kind of what we had hoped for, but at the very least, we didn't see a dramatic decrease in the Fingolamod treatment in terms of CD8 T cells. So if I come to the conclusion slide now, the last slide, the way we're sort of thinking about these and future experiments are that we can use this sort of mouse modeling therapeutic approach to sort of try and hopefully ultimately tailor patient therapies, and as we heard before, ideally combination therapies to different specific genotypes of CCICC. So at least in this model, we were able to show in previous studies as well that this mutant background is strongly sensitive to everolimus and to fingolimod, more moderately sensitive to sunitinib and acroflavin, and completely resistant to HIF2 inhibition. And what I haven't shown you is it's also resistant to checkpoint therapy, as well as various combinations. So at face value, we might suggest from this that patients who are failing checkpoint therapy and potentially in future failing HIF2 inhibitor therapy might potentially be treated with sphingosine pathway inhibitors or even dual HIF inhibitors. And we're about to start testing actually fingolimod in advanced CCICC patients in a small cohort. And finally, as you all know, it's been very exciting in recent years that many other models of different genotypes of CCICC have been developed, and Bill Kalin showed us some very promising new approaches, maybe for more high throughput modeling. And it'll be interesting to use a similar approach to see if we can tailor specific therapies in the mouse, at least, to the genotypes of those different models. So the final slide is the acknowledgement slide, where I'd really like to thank all of the members of the labs actually the labs in Zurich and Freiburg now, who've worked on this over a long period of time. Sabine Harlander did basically most of the mouse studies. The analyses were done here, taken over by Reuven Höflin, in collaboration with many of the great colleagues here, specifically those labelled in red. And thanks, of course, to all of the funding sources, not least of all the VHL Alliance, who supported this project at an early stage. So thank you for your attention. I apologise for the technical problems and happy to take any questions.
Thank you, Ian. Great talk. Fantastic work. We have a couple questions here in the Q&A thing. Both are from Ching Shang. He asked a technical question, should be quick. It looks like the HIF2 alpha immunohistochemistry staining showed a diffused staining pattern in the VPR triple knockout. Is that an antibody limitation, do you think, or is that some of the HIF2 alpha is actually localizing in the cytoplasm or elsewhere in the cell?
Yeah, I don't know. I think the quality of the immunohistochemistry doesn't necessarily tell us that. We had an enormous problem. I think if you read carefully in the paper, we tested something like 7 different antibodies to try and find one that worked. We're very confident that the nuclear staining is real, but I wouldn't want to say anything based on the weak staining that we're able to get there.
His other question is, it's interesting that VHL loss combined with HIF1 alpha, HIF2 alpha loss further increased SPHK1 levels. I'm glad that it was validated, their finding with regard to VHL regulation on SPHK1 in HIF-dependent manner. Would you expect that combining HIF2 alpha inhibitor together with an SPHK1 inhibitor would work synergistically? It's a great question. Yeah.
Excellent question. We don't know. I think the key to understanding that question will probably be to try and understand how much of the therapeutic effects of sphingosine kinase 1 are actually running through HIF1 or HIF2 reduction of protein expression. We don't know the answer to that yet. It would certainly make sense if the sphingosine effects are independent of HIF levels, then it would make sense to combine, two different inhibitors that work via different mechanisms. I completely agree, but I think these are the questions we actually now have to go and ask. Yeah, thanks for that suggestion.
Right, and I was also just curious myself about what do you think? Do you think it's just the metabolic maturity of the renal cell carcinoma that causes the clearness, as you called it, the clear cellness?
I mean, I think it's certainly clear that the clear cell phenotype is due to the accumulation of lipids and of cholesterol. And we haven't gone and looked in a lot of detail at metabolic, or lipidomic sort of profiling of the different genotypes. But there are different paths to accumulating lipids, and so I think probably that HIF1 and HIF2 are affecting different aspects of that. What was interesting was that HIF2 seems to affect the transport and the storage of the lipids, whereas HIF1 may be more affecting the pathways that would lead to sort of de novo lipid formation or fatty acid formation.
Okay, great. Thank you so much for this informative talk. I don't see any other questions there, so I'm going to